We're ready now to move on to chapter 6 in our microbiology textbook for our introductory microbiology course. And this chapter is about microbial growth. We'll learn about a little bit about how microbes grow and what they need to grow, and how we can learn a little bit about their identification in the laboratory by how they grow, and some various tricks we have for that. There's a little quick micrograph of a very common um, microorganism that can cause disease in the hospital setting, Serratia marcescens. I think that came up in our last chapter too. So, microbial growth. We're talking in this case about multiplication of numbers, not microbes getting bigger and bigger. So that's what we mean by growth, just to clarify. We have a population of cells that's increasing. Sometimes if we're growing microbes on some culture media in the labs, we can observe them growing. We'll produce or observe the appearance of colonies of microbes, colonies of bacteria, and so forth. And we'll look more at that in a little bit. So let's talk about some physical requirements for microbes to grow. There are some conditions under which they grow well and those which they don't. And we'll talk about what they need to eat. Here's a interesting graphical representation of the temperature ranges in which different classes of microbes can grow. For example, mesophiles. Let's look at that one. It's this represented by this red line that encompasses temperatures ranging from 10 degrees Celsius up to 50 degrees Celsius. Mesophiles are the most common types of bacteria that cause disease in humans, and their optimal growth temperature is, not surprisingly, the temperature, I shouldn't say just disease causing, but bacteria that thrive on or in the human body. They have an optimal growth temperature of human body temperature, 37 degrees Celsius. That's what they're adapted to grow at. That's where they've been growing for generations, untold. Mesophiles. Thermophiles are bacteria that are adapted to growing at higher temperatures, and they can survive in much higher temperatures than mesophiles can. And then hyperthermophiles are ones that really need a good hot temperature to do what they do. Interestingly, if you go to a hot spring like Old Faithful, if you dig around where the water's coming out of the ground at upwards of 100 degrees Celsius, you'll find living microorganisms there, hyperthermophiles, and some very important methods in molecular biology depend on some enzymes that are produced by hyperthermophiles. Psychotrophs are bacteria that can grow at much chillier temperatures, right? Some spoilage bacteria, for example, can grow in the refrigerator, unfortunately. Psychotrophs. Try to chill them to slow them down. Most of the time when you chill microorganisms, they don't grow. They grow more slowly at the slower temperature, at least. These guys are better at growing in cool temperatures. Psychrophiles are ones that actually like the colder temperatures, almost need the colder temperatures to grow. So there's kind of the extreme guys on that cold end of the spectrum. There's a picture of a vent, a hot spring, essentially, if you will, at the bottom of the ocean, where water that's been heated by magma down deep in the crust of the earth coming to the surface is very hot. And there's a bunch of microorganisms growing right there where that hot water is coming out. Mesophiles are the bacteria that we're going to be most interested in because they're the ones that are going to be colonizing our bodies, either for good or for bad. Psychotrophs are of some interest because they may grow under conditions at which we try to preserve our food and prevent spoilage, and prevent disease-causing bacteria from growing. Excuse me. And so those are the temperature ranges for those two types of things. Here's a little wake-up call maybe. If you take a container full of food that contains bacteria, in this case we're talking about Bacillus cereus, which is a microorganism that not commonly but can be found in fried rice preparations. If you stick that container in the refrigerator and see how long it takes to come down to fridge temperature. Let's say we take a nice warm bowl of rice at 70 degrees Celsius, we heated it up, and then we're done with it. We stuck it in the fridge. how long it takes maybe 3 hours for it to chill down to fridge temperature approximately. And in the meantime, for a couple of hours, it's at the temperature of that food is the optimum growth temperature for the bacteria, the pathogenic bacteria we're not interested in eating. So if you put a larger container that has a larger mass or volume of food in it, now look how long it takes. In 8 hours, it's still in the optimum growth range for the bacteria. So you can start to see some wisdom that you can acquire about how to store food um, in the refrigerator. Make sure you don't put it into very, very large containers and so that it can chill a lot faster. Physical requirements uh, in terms of pH. We just look at temperature. Most microorganisms have a pH range that they're adapted to. Most bacteria grow around neutral pH. Neutral we would call 7.4. That's the pH of our internal body fluids and so forth. There are some regions of the body that are different pHs, but that's a typical um, bacterial growth range in terms of pH. Molds and yeast tend to grow at more acidic temperature pHs. I'm sorry. There are microorganisms that grow in very, very acidic environments indeed. Acidophiles can grow in acidic water. Say, for example, we just talked about the hot springs. Sometimes those waters are quite acidic from coming up through various mineral materials which they extract as they're passing by and can be quite acidic. Osmotic pressure. If you place bacteria in a hypertonic solution, we talked about tonicity last chapter, and a hypertonic solution is one that causes water to leave the bacterium by osmosis. It's more concentrated than the cytoplasm of the bacteria, and when the bacteria shrink down in this way as water is leaving, that's called plasmolysis, and many microorganisms can't grow. They can maybe survive that process, but they can't grow in those conditions. So you can inhibit the growth of bacteria by placing them in hyperosmotic solutions. Right? I mentioned it before, putting fruit in syrup or using salt on meat to preserve it because the spoilage bacteria can't really grow very well under those high osmotic conditions. Here's a picture of plasmolysis happening. The bacterium is shrinking down. You can see its cell membrane and all of its cytoplasmic contents are shriveling right up as water leaves by osmosis. There are microorganisms, facultative halophiles, that can grow even if it's very, very high osmotic pressure or salty conditions. Halophiles refers to salt conditions. Staphylococcus species that grow on the surface of your skin. It's very dry and salty on the surface of your skin. When sweat comes out and it dries up, what's left behind is the salt that was in that sweat, and so the surface of your skin is a very salty, dry desert essentially. But some microbes like staph bacteria can survive that. They're not exactly thriving, but they can survive out there on the surface of your skin, so they represent some of the more common bacteria there. This picture is talking about plasmolysis above, but the lower part of this text down here doesn't have anything to do with plasmolysis. It's talking about the fact that bacteria don't need to regulate their fluid volume. Yes, you can put bacteria in a hypertonic solution, but usually bacteria find themselves in a hypotonic environment, which means water will be trying to diffuse or moving into the bacterial cell by osmosis because there's more concentrated solute inside the cell, a lower water concentration. But bacteria have a cell wall so that it doesn't really matter. The water there's osmotic pressure, and so there's pressure that builds up inside the cell wall of the bacteria, but that's okay. It's strong enough to handle that, and so the bacterium just lives like a tight little bullet of high pressure fluid in there because of osmosis. If you do something to disrupt the cell wall, as we'll learn more about in the next chapter when we talk more about specific types of antibiotics, if you have some lysozyme or an antibiotic that interferes with the synthesis of the cell wall, the cell very well may burst because of the osmotic pressure. Chemical requirements for microbes to grow. Carbon. All organisms need some source of carbon so they can build stuff. They need to build proteins. nucleic acids, various types of sugars for metabolism, and and lipids of various types. So they got to have carbon. We talked about already before about chemoheterotrophs, for example, use carbon as their source of energy and also external sources of carbon for building stuff out of. So they use organic carbon sources. They have to get some carbon molecules. They have to maybe digest some proteins outside the cell and bring in the amino acids. Autotrophs use CO2. They can fix CO2. That means take CO2s and attach them covalently together to form a 6-carbon, ultimately a 6-carbon moiety, and make sugar out of it. So that's how autotrophs get their carbon. How about nitrogen? There's nitrogen in amino acids. This is the amine. An amine group in organic chemistry is a nitrogen-containing organic group. So amino acids have nitrogen. Um, um, Nucleotide bases have nitrogen, nucleic acids. So we need plenty of nitrogen in order for bacteria to do what they do, for living cells to do what they do. Some organisms can use ammonia or nitrate. But some organisms, amazingly, almost magically, can take nitrogen out of the air. 80% of the atmosphere, or a little less, is nitrogen gas. All that nitrogen out there and nothing you can do with it, because most organisms can't do anything with all that nitrogen, but some can. They can do what's called nitrogen fixation and convert it into ammonia or nitrate. And that's a magic trick, because then all the other organisms in the world rely on those few microorganisms that can fix nitrogen. All the animals, all the plants need that nitrogen in a form that they can use, and it all comes from a few nitrogen-fixing bacteria. Pretty cool. Sulfur. Certain amino acids have sulfur in part of their side chain, so you have to have sulfur to make those amino acids. Some bacteria, as we've already seen, can use sulfate ion or hydrogen sulfide as a source of energy or electrons. Phosphorus is found in DNA and RNA in those nucleic acids. The backbone is pentose sugars and phosphates. Phosphorus, inorganic phosphate gets strung along in those chains. So you need phosphorus, which is the atom that's bounded to oxygen to make phosphates. So we have phosphorus and oxygens. That's a phosphate ion, and that's again used to build nucleic acids and ATP and all those kinds of things. And there are other trace elements that are needed to be in the soil or in the environment of bacteria for them to grow. As is true of most cells, need a few really odd kinds of micronutrients, tiny amounts of certain things that are involved in making enzymes actually do their thing. Cofactors for enzymes are sometimes trace elements. That would be like selenium, for example. Like, what? Yep, need it for certain enzymes. Molybdenum. All right, here's another topic. How microorganisms deal with oxygen. And these cartoon drawings represent 5 different classifications of microorganisms according to how they handle oxygen or deal with oxygen. In this first left-handmost cartoon, where this cartoon of some growing bacteria represents obligate aerobes. That means they have to have oxygen in order to grow. They can only do aerobic respiration. They can't ferment, so they've got to have oxygen. And here you can see we've put them, here's a test tube full of agar, a gel, or if it's a liquid medium, it's held very, very still. And we inoculate the surface, or inoculate the whole thing, actually, doesn't matter. We inoculate this agar with some bacteria, and there's air inside this part containing 20% oxygen. The only place the microbes can grow is here at the surface where there's oxygen diffusing into the agar. So that's obligate aerobes, got to have oxygen. Facultative anaerobes. And this word facultative, now we saw it come up a little bit ago with facultative halophiles. Facultative anaerobes are ones that can grow anaerobically. They're able to still grow under anaerobic conditions. So again, here we've inoculated this whole agar tube, And you see there's growth all throughout the agar. These bacteria can grow in the absence of oxygen. Oxygen would not be able to diffuse that far down into the agar. But look at the surface. They grow better in the presence of the oxygen. So facultative anaerobes can grow anaerobically, but they grow much better aerobically. They have all the equipment to do aerobic respiration and get the 34 ATPs per glucose. Obligate anaerobes cannot tolerate the presence of oxygen. Oxygen is nasty, toxic stuff. When there's oxygen around, toxic oxygen species or forms will occur, which will damage and destroy cells that don't have a way to combat it. Isn't that strange? You think of oxygen as being so life-giving, but it actually is very nasty, toxic stuff. Without a remedy, cells can't live in the environment of oxygen. All right, so we have obligate anaerobes are just such organisms. They cannot stand oxygen. And so we see in this tube, the only place we see the microorganisms growing is way down here at the bottom where there's absolutely no oxygen. It hasn't diffused that far down. Aerotolerant anaerobes. Now these are anaerobes, much like facultative anaerobes, that can grow without oxygen, but they can't utilize oxygen. They gain no benefit from being up here near the surface in the presence of oxygen, so they grow at the same rate whether there's oxygen present or not. They have a way of dealing with the toxic forms of oxygen, but they can't utilize it. It does them no good. So there's an even distribution of growth throughout this agar tube. And then finally, microaerophiles. Microaerophiles need oxygen, but they need a low level of oxygen. They can't tolerate high concentrations of oxygen like in the atmosphere, and they can't grow anaerobically. They need a small amount of oxygen, just the right amount, like the Goldilocks of bacteria, right? And they're called microaerophiles. So you need to know those, be able to know these classifications. What would if I asked you what kind of bacteria is growing in this tube here, what would you call them, and why does the growth look the way it does? Make sure you can explain that. So yeah, oxygen produces toxic species. Here we have superoxide radicals and anions. You see the anion is that negative charge there. That radical, there's an unpaired electron there, which is a very, very reactive thing, and the oxygen will go around chemically reacting with stuff and causing damage to DNA, damage to proteins, can kill cells. So most cells that can live in the presence of oxygen have a gene to produce a gene product called superoxide dismutase. It's an enzyme that will take these superoxide radicals and convert them to hydrogen peroxide. And hydrogen peroxide is not all that great. But also, catalase may be another gene that these organisms have at their disposal, which will take the hydrogen peroxide and harmlessly change it into water and oxygen. And so we've taken something extra nasty, and a couple of different enzymes present in the cell can get rid of it for us. Peroxidase is another enzyme that some microbes have to destroy hydrogen peroxide and turn it into water. So that's what cells have to be able to exist in the presence of oxygen. Some cells have antioxidants present in the cell that can get rid of hydroxyl radicals, for example. So instead of having an enzymatic process, they just have antioxidant proteins that will mop up these oxygen or hydroxyl radicals so that they don't go around damaging the cell. So like having a sacrificial protein present there to get rid of it. Organic growth factors, organic compounds. Some microorganisms can make almost anything, right? Maybe they just fix CO2 and form sugar, and now they can take that and convert it to every single thing they need. But most organisms have a few things that they can't synthesize, and they have to get it from outside. So they have to obtain some organic compounds from the environment that represent a group of substances they need, but they can't synthesize it. So most organisms have a sort of a short list of things that need to be provided for them to be able to grow. And we as microbiologists will provide that for those organisms to give them what they need so we can get them to grow in the laboratory. Oh, there I go. I disappeared again. It's all right. Let's talk about biofilms briefly. We've brought it up before and given a quick definition, but when bacteria grow, and begin to accumulate in a particular microenvironment. They recognize the presence of each other. They give off signals that they start to each realize that they're in a very dense population of bacteria, and they start secreting some polysaccharides, some sticky, slimy polysaccharides that begin to sort of cover over the whole population of microbes and sort of allow them to ensconce themselves down in the slime. So quorum sensing is that ability of microorganisms to recognize how many other examples are around them, what's the population density becoming, and they start secreting stuff that we call biofilms. We call it that because they'll form this layer of material that in which they're housed and that protects them from the immune system, protects them from antibiotics, protects them from disinfectants. So they're much harder to kill in the presence of a biofilm. And that's a big problem in the medical setting, because when there's microorganisms on the surface of, say, something that's been inserted into someone's body, it can allow the growth of bacteria in a biofilm. It becomes very, very hard to, or impossible even, to get rid of. So the bacteria and the biofilms share nutrients, and they are sheltered from antibodies, right? The immune system, immune cells, disinfectants, and antibiotics, as I mentioned. Should have waited for that slide, right? There's a picture of a biofilm forming. Indwelling catheter, what in the world is that? Oftentimes, a plastic tube will have to be inserted into the vascular system or into some organ of the body in order to retrieve some blood samples periodically, or to introduce something into the body for purposes of hemodialysis, for feeding tubes, for getting food into the body, any number of things. And these catheters that are in the body for long periods of time, even catheters in the urinary tract to help to extract urine out of the bladder, they can have a biofilm may grow on there, and the bacteria that are protected in that biofilm may cause disease. Here's an example. Patients with indwelling catheters in the vascular system received heparin. Heparin was used as an anticoagulant to prevent blood clotting for some reason. Maybe there was some excessive clotting happening. It turned out the heparin was contaminated. But when the microbiologists examined the heparin, the contamination was so low, so few bacterial cells, that it would have never been enough to cause disease. The immune system would have gotten rid of it in no time flat and eradicated it. But it turned out that people did develop infections, and look how long it took. So just a couple of bacteria managed to hang on to that catheter and begin to grow into a larger number and secrete some of that biofilm material. And eventually, a very significant population of cells began to grow in there and cause disease in the patient. How about culture media? Now we're talking about methods that are used in the microbiology lab to grow bacteria, either to identify them or to use them as tools for various purposes in molecular biology. So culture medium is what you provide for the bacteria to be able to grow. It's going to have all the nutrients that are needed, the carbon source, various chemicals that the bacterium can't synthesize, and so forth, nitrogen and sulfur and phosphorus. Agar is a gelatinous stuff. It's like jelly, basically, and it comes from certain types of seaweed. And it's sugars, polymers of sugars, polysaccharides from the seaweed that when you heat them up, they melt. And you can pour the melted polysaccharides that have been purified from the seaweed into a petri dish, let it cool off, and it'll solidify into a gel, and it makes an ideal substrate for or support for microbes to grow on in the laboratory. So we typically grow microbes either in liquid broth or on agar gels. Sterile, the word sterile means there are no living microbes. If you sterilize something, right, if you want to grow some microbes, first you got to get some agar and put some culture media in it that's going to supply all the nutrients needed. And you got to sterilize it somehow so there's no bacteria in it to start with, so we can add bacteria that we want to grow. We want to grow something specific, right? So we're going to add something to it then. But first it has to be sterile, have nothing living in it. We're going to then take that sterile medium and inoculate it with the bacteria that we want to grow, the microorganisms we want to grow. It doesn't have to be bacteria necessarily. Inoculation, or the inoculum, is the stuff that we actually place on the sterile medium, the inoculum. And that in the process, the verb is called inoculate. All right, so the culture is the growing microorganisms. Once we inoculate that sterile medium, now we're going to have a culture, hopefully with any luck, microbes growing on there. So we've got agars, a polysaccharide purified from certain types of bacteria that if you heat it up, it'll melt. You can do it over and over. And then when you let it cool down, it solidifies back into a gel, allows us to pour it into glass tubes, pour it into petri dishes, and any number of things. Let's talk about different types of media. We can have chemically defined media in which we know precisely the concentration of every constituent in the media. Sometimes that's very useful to do that in terms of identifying what can and can't grow on it. Oftentimes, though, we just want to get stuff to grow. We have a complex media in which we take a bunch of nutrients from either yeast or meat or decomposing plants or something like that and place it in with the agar. And now we have a wonderful mixture of virtually every nutrient that any bacteria could possibly need. And one particular type is called nutrient broth or nutrient agar. So what is that? Well, first let's look at a chemically defined medium. Here's an example of a chemically defined medium. It has glucose, that's a discrete molecule, hexose sugar, ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, and water. Those are the only things in this medium, and they're in there in very specific amounts, and E. coli can grow on that. All it needs is glucose. It needs that carbon source, and it can produce everything else it needs. What an amazing metabolic powerhouse it is to be able to do that. Well, nutrient agar is a complex medium in agar that allows virtually any bacteria to grow on there. Not all bacteria have all the tricks that E. coli do. Not all bacteria can produce all the biomolecules that they need from glucose as a starting material. So we'll take some partially digested protein. Just take a bunch of protein from whatever source, whether it may be milk, and add some enzymes that kind of probably break it down. Mix that with some extract from meat, some beef extract. We'll have a bunch of amino acids and small peptides all in it. Add some salt, some agar to make the gel, and boom, we have a very rich media that can grow virtually anything. It's called nutrient agar. There's all kinds of media. There are dozens of different types of specialized media, just so you know, but we're not going to worry about that right this minute. Reducing media is a type of media that removes oxygen from it so that by virtue of certain chemicals that are included so that you can grow obligate anaerobes. Remember, obligate anaerobes can't stand the presence of oxygen. They don't have catalase or superoxide dismutase, so they'll be killed in the presence of oxygen. So we got to get rid of it. So we'll produce a medium that has all the nutrients needed, but it'll also have first of all, we'll heat it to drive off any oxygen that's dissolved in the liquid, and then we'll also add some thioglycolate, for example, that will capture the last remaining bits of oxygen out of the media, and that allows those obligate anaerobes to grow, and then we can study them in the laboratory. Here's a little anaerobic chamber, and it's going to be a chamber in which there's no oxygen in here at all. These palladium catalyst pellets allow for reacting with oxygen so that it's taken out of the atmosphere. Is another way of getting rid of oxygen. Here's some agar plates with that agar medium in there, lots of nutrients in there, and we've inoculated it with our bacteria of interest. Now it's going to grow on these plates. One interesting point to make, I'm sure you've already learned in the laboratory, but in a microbiology lab, you incubate your samples, your agar plates, in this inverted fashion. The agar is on the bottom of the plate and it's turned upside down, because condensation will form on the lid of the plate. These are plastic petri dishes, and the lid is down here of this top plate, and condensation will typically form on the plate. You don't want that to be on the agar, because of the condensation, it will start droplets of water flowing around in there. It'll cause cross-contamination of different colonies that form there. People that study anaerobic bacteria in major experiments need some facility to grow them on a regular basis. They can't get away with just this little anaerobic chamber. Here's an actual hood for working with microorganisms and various equipment inside of a chamber that has no oxygen. Typically, hydrogen gas has been used to produce these kinds of chambers, which may seem crazy, and it is a little bit, and there's probably very good reasons why hydrogen to this very day is used for these kind of anaerobic chambers, but hydrogen will explosively react with oxygen in the atmosphere unless you're very, very careful. So it's a very kind of dangerous um, um, gambit to be working on anaerobic bacteria research using one of these anaerobic chambers. But you can see that the scientist has to slip their arms into these arm ports to be able to reach inside the chamber and work on stuff, manipulate things physically, but without ever actually introducing any of the atmosphere from the room into there. Here's a little port in which something can be placed in here from the room, evacuate all of the atmosphere out of here, replace it with the hydrogen gas from the chamber, and then be able to open a little door inside here and retrieve whatever substances, plates of media, who knows, into the chamber. I will tell you a quick story. I used to work at a research institute in Cleveland, in the Case Western Reserve University, and one morning when we came in to go to work, our slide card access wouldn't work, and security had locked down the building because there had been a big explosion early in the morning, and one of these hoods just blew right up, and glass shrapnel flew everywhere. A couple of people working in the lab were injured and knocked unconscious and so forth by the shock wave of the explosion. So it really does happen by grace of God. The postdoctoral fellow who was working in that hood had left the hood and went around the corner in the lab around a stone, a cement block wall into a hallway just around the corner from the hood when it exploded. So they were kind of shook up a bit, but didn't get glass impregnated into their body from the explosion. Quite crazy. Anyway, capnophiles are microorganisms that need a lot of carbon dioxide, amazingly, in order to grow. And so we may place them in a container that contains a lot of CO2. Chemical packets can be obtained that by chemical reactions, once you start them, you stick them into the container along with the petri dishes of agar, and CO2 is released and makes those things happy in there. A candle jar has you can stick an actual literal candle inside the jar, which will burn oxygen in the air in the jar and give off CO2. Another way you can produce a high concentration of CO2 in the atmosphere. A whole nother topic. When you study microorganisms or handle them in the laboratory, you have to be careful that you don't make yourself sick or make anyone else sick, either in the community or other workers in the environment. And so the Centers for Disease Control has studied all different types of microorganisms and sort of categorized them according to their risk for causing disease. How bad will the disease be and how easy is it to treat? And they've created these so-called biosafety levels, which are recommended procedures or precautions to take with various microorganisms to prevent undesired outcomes. So biosafety level 1 is no special precautions are needed. Biosafety level 2 is by far the most common, and it means that you're going to use personal protective equipment, PPEs, such as a lab coat and gloves, eye protection, and you're going to work inside of a biosafety cabinet so that all of the microbes you're working with are trapped inside this cabinet and not freely going out into the room and so forth. And also nothing from the room is going in there and contaminating your experiment, but that's biosafety level 2. And there are lots and lots of organisms on a list of things that are handled at biosafety level 2, usually things that cause very modestly dangerous diseases for which there are very, very effective treatments available. But yeah, we don't really want everyone to get sick and have to be treated by these things, for these things. Biosafety level 3, much more virulent or disease-causing organisms that maybe it's a little harder to treat them. Antibiotics are more difficult to treat with and so forth. So biosafety level 3 involves working in a special facility in the research institution in which there's a negative pressure of air, meaning there's air being sucked into the facility from every direction so that none of the microorganisms, these horrible disease-causing agents, could ever get out into the rest of the university system from the room. There's fresh air that's filtered. HEPA filtered air is coming in from in every direction. HEPA filter means removing all microbes by forcing the air through very, very tiny filters and to prevent anything escaping from the research lab. And biosafety level 4 is the highest level of safety of all for handling microorganisms for which there is no treatment. So if you get infected with this thing, you're probably going to succumb. So be extremely careful. Anyway, I belabored that enough. And here you see people suited up for biosafety level 4 research, handling some microbes that cause very, very terrible diseases for which there is no good treatment. Not only is there negative pressure in the laboratory, there's positive pressure in these suits so that no microbes can kind of get onto the person's body and get in there and start moving around on their body surface, because there's HEPA filtered air coming into the suit and blowing outward from every direction to make sure nothing can go in from the area of the hood where they're working. All right, that's the end of part 1 for chapter 6. We'll pick up again for part 2 and learn a few more things about microbial growth, different specific types of media that are used to identify microorganisms and so forth. So join me for part 2 next time.